So it's very nice to be here. What I will try to do today is to talk about one of my favorite topics. I guess that's a pretty nerdy thing to say about antibody validation, but it is something that I want to convince you that this is not easy. There is no such thing as a good and a bad antibody, and that's what I want you to sort of learn during my during the first 30 minutes here. Just want to make two disclaimers. I'm the co-founder of Atlas Antibodies and Antibodypedia, and just wanted to mention that at the start. I thought I would go through 5 different topics today. First, a short update on the Protein Atlas. Some considerations when it comes to validation of antibodies. Then talk about the proposal for antibody validation that I was sharing, and then how we have used this. And I will use Western blot as the sort of example here. And then how we have implemented this in the Protein Atlas. So just to give you first a glimpse and introduction to the Protein Atlas, I hope you all know about it. But it's an international effort to try to map all the human proteins using antibodies. We have made more than 55,000 antibodies in-house. We immunized about 7,000 rabbits per year for many, many years. But we also used antibodies from more than 50 providers, so it's been a massive effort. We decided to have all the data in an open access format. And right now, we're focusing a lot on antibody validation. That's why I'm so excited about this meeting. We have 3 parts of the Protein Atlas today. The Tissue Atlas, that was the first one, came in 2015. We published it in Science. It has more than 2,500 citations now, so we're quite happy about that. There's about 3 citations every day, which is kind of nice. With the antibodies, it allows you to do single-cell resolution, as you all know. You can look with immunohistochemistry on the context of neighboring cells and so on. So that means that we can zoom into tissues like, for example, kidney, see where are the proteins, are they in the glomerulus, in the tubuli, and so on. And then publish a lot of images. So we have published more than 10 million images now. Each one of them has been annotated by a pathologist, and we use pathologists in India. The second flagship paper from us was the Cell Atlas. This is about a subcellular profiling using antibodies. It should not be mixed up with the Human Cell Atlas initiative, which is focusing on RNA and genomics. This is using antibodies. And we published then a paper in Science about 1 year ago, I guess, which then shows you the sort of single-cell variations of cells. We have some beautiful examples here. This is, I just show you the rods and rings. That is this peculiar structure that you can see in some cells. You can also see with one image a lot of things about the function of proteins, such as this protein, which is, of course, as you can see here, let's see, you can see that it's interacting with the centromere, but when the cell has divided, it actually moves to the cytokinetic bridge to the two dividing cells. So and then, of course, in most cells, it's not expressed at all, which are not dividing. We see a lot of proteins with cell-to-cell variation. So this is just one example where you have this protein, which is produced during this S phase here. The third sort of flagship paper from our group came in the end of last year, about a year ago actually, and that is the Pathology Atlas. It's very different. Here we are asking what is the proteins that actually affects the survival of patients. And I will not talk so much about this because this didn't involve very much antibodies. So basically, the Protein Atlas now contains 3 separate parts: the Tissue Atlas, the Cell Atlas, and the Pathology Atlas. And a lot of the focus in these 3 atlases is now on the antibody validation. And we will release a version 19 in November, 15th of November. And then there will be a lot of new validations done. But what we're really excited about right now is 2 major launches that we hope to do next year, which we have demo versions of them right now. And that is a blood atlas, where we are looking at both cells and proteins in human blood and trying to characterize what proteins are involved. And this is very much using antibodies. And then we also have a brain atlas where to look more in detail in human brain, but also mouse brain and pig brain, and comparing, again, using antibodies, but also using transcriptomics. So this is very exciting for us to add these 2. And both the blood and the brain is not really covered very much in the first 3 launches of the Protein Atlas. So then I want to move a little bit about antibody validation then. Already back in 2008, we published a paper in Molecular Cellular Proteomics where we looked at antibodies from commercial providers. And we showed that these 30 providers had sort of mixed success rates when we were using them in our applications. And about 30%, the average success rate was about 30% of an antibody working in our hands. So this was a little bit alarming, and we published that, but there's also quite a lot of other literature that was published about anti-validation and why this is very important for the community. Then there were some rather alarming editorials, and I'm sure some of you recognize these, especially from Nature, calling and blaming the antibodies for a lot of misinformation in literature. So with this, then, we decided then to start a portal for antibodies. We call it Antibodypedia. The idea was to allow researchers to compare validation data for different antibodies to a target protein, and to have this in an open access way with no charge. We just released a new version today, actually, of this, and it allows you to search for antibodies for a particular target or transcription factors and so on, and for a given application, which is kind of nice, so you do the search in an application-specific manner. The Antibodypedia has been a fantastic success in one way. We now have 3 million reviewed antibodies to human proteins. 82 antibody providers are giving information to it. We cover 94% of all the human protein-coded genes. But maybe the most impressive is that we now have 1.4 million validation experiments that are actually in Antibodypedia. So we're very happy about this. Let's see now. Yes, okay. Just to give you some statistics, because this is mind-boggling for me. Because the reason in the Protein Atlas we made so much antibodies ourselves was that there was antibodies lacking, we thought. But for sure now, in average now, we have 151 antibodies to every human protein, which is kind of amazing. If you look at some proteins, EGFR, for example, in Antibodypedia, there's 6,500 antibodies to this target. It's almost crazy, isn't it? And if you look at the statistics, for 88% of the proteins or the protein-coded genes, there is more than 10 antibodies in Antibodypedia. And there's only 7% where it's less than 10. So that means then that if you're interested in this particular protein class, blood groups, 100% of them are covered by antibodies, nuclear receptors, 100%, and so on. And even if you go down to the protein class predicted proteins, which no one has seen, there is actually quite a lot of antibodies to them. More than 98% of them are covered with antibodies. It's quite astonishing. And you should sort of note here the y-axis. So then, of course, the problem is not the availability of antibodies. The problem is what is a good and what is a bad antibody. And I hope that at least you come from my lecture today and understand there is no such things as a good and a bad antibody. There might be bad antibodies, but good antibodies is very much context and application dependent, which was outlined by Baker in Nature some years ago. So I give you a few examples because I think it's good to show. So this is an antibody towards this very famous protein. I have no idea what it's doing, but it's not supposed to be in cerebral cortex, kidney, and liver because there's no RNA in these tissues. And you can see, and then there should be some protein in the testes. You can see a lot of staining in cerebral cortex, kidney, and liver. So I think most people would say this is probably not a very good antibody. Bad. Then we have another antibody, and it's staining the same tissues, and you can see that it doesn't stain at all the brain, the kidney, and the liver, and it stains very nicely the testes. So it is obviously a good antibody. And actually, we show also with the Western blots and so on, this is a very good specific antibody. The only problem with this is that it's the same antibody. So it's just that it has been diluted 50 times from here to here. So basically, what it shows is that this is a good antibody, but if you do it in the wrong context, it becomes a bad antibody. And I think it's very important to realize that it's a lot depending on the researcher what is good and bad. I also show you another example, what we are doing for all our Protein Atlas generated antibodies, and we produce 55,000. We put them into microarrays with a target in 2 places, and then we score if they are recognizing the target in the background of hundreds and hundreds of other proteins. And here is an example of an antibody that recognized the target, and it's completely blank for all the others. And actually, out of our 55,000 that we generated, more than 50,000 of them were actually as this, very, very specific for the target. So is it any problem? Well, we then moved them into immunohistochemistry, and less than 30% of them actually works in immunohistochemistry. So obviously, it's a good antibody, but it doesn't work. So it's also a bad antibody. And I think you have to realize that this is application dependent. And how can this be? Well, it's very simple. And the reason, of course, is that when you move into the most common applications in life science, you treat the samples very differently, and the proteins are being treated very differently. So Western blots, everybody knows that you use SDS, which is very harsh to the proteins. For the immunohistochemistry, it's almost extreme because you add formalin to your protein, and then you boil it or even have it at 120 degrees. And obviously, you completely destroy the protein's native structure. It's called epitope retrieval by the pathologists, but it's really epitope destroying in a way. But that's the way it's done. So, and then in other applications, like flow sorting, you are actually looking at the proteins in a native form. So depending on the application, the protein will be very, very different and therefore be looked by the antibody very differently. This is a very busy slide, but there are two very good papers from our group. I know I'm biased, but I think they are fantastic, actually. And I thought I would go through this one from Protein Science, which probably no one has read, but it's a very good piece of work. Again, unbiased. So this is a polyclonal antibody to a target. What we've done then is that we have done a very elaborate chromatography and actually eluted up. First, we determined the epitopes using arrays, and then we actually eluted out the epitope-specific antibodies, and then we looked if these epitope-specific antibodies, a little bit like a monoclonal then, artificial monoclonal if they work in Western blot immunohistochemistry and immunofluorescence. And what you can see, for example, for this epitope here, it works beautifully in both Western blot immunohistochemistry and immunofluorescence. But you can see the next epitope next to it, it works actually in Western blot and immunohistochemistry, but it doesn't work in immunohistochemistry. And then you have, even worse, this guy here, which doesn't work in Western blot immunohistochemistry and immunocytochemistry. So what you can see here is that depending on what epitope it is showing, it works or not works. And obviously this is because if the epitope is still there. So this is the reason why we in the Protein Atlas have worked very much with polyclonals, and we are very happy about polyclonals. The nice thing about polyclonals for research application is that they have several epitopes, and we estimate normally that we have 3 to 5 major epitopes per polyclonal. Monoclonal antibodies are wonderful in many ways, but it's a single epitope, and of course that means then that they are not very good when you want to have functionality across applications, because you depend that that epitope stays when you move from native to very denaturated proteins. And also, when you do variations of the sample, you depend much more when you have a monoclonal. The good thing about monoclonals, you all know this, is of course that there is no batch-to-batch variation. So obviously, if you use polyclonals, you have to be very careful when you make a new batch that you validate it the same way as you did in the first case. So I'm not going to go into the details here, but basically there are 3 issues then for reliable antibody use. It's the target binding, cross-reactivity, and reproducibility. So with this in mind then, and how we're doing on time here, I still have 12 minutes, I think. So with this in mind, we had a very nice initiative actually from Thermo and Matt Baker, which is in the audience, to start an independent working group consisting of people both from Europe, from Asia, and from America, which I was sharing. And we worked for about 2 years trying to identify ways where you can formally validate antibodies. And it was a very interesting 1.5 years. Basically, when we started, we were discussing 2 main principles to do validation of antibodies. One would be to try to find a scoring, some sort of scoring list that you can actually score every antibody according to some parameters. And basically then in the end you will have an antibody for a target which is the best one and the second best, and you would be able to pick the winner with the highest score. You kind of understand that I hate this concept, because the concept sort of says that a good antibody is always a good antibody. And it will really be rather bad for the community if we go for this. Because a person who doesn't actually understands the complexity of using antibodies will, of course, pick the antibody with the highest score. So we didn't go for this one. Instead, we said, let's try to validate antibodies with predefined criteria in a particular application. And after a lot of discussions, we formulated 5 pillars, and we hope that you will add more pillars in the future, but so far no one has suggested a 6th one. The idea here is that you should be able to use these validations without any prior knowledge of the target, because a lot of these targets are really not that known. And I will go through them one by one in our settings just to give you an idea how they are working. And this was published in Nature Methods about 2 years ago. So for us, these 5 pillars are now used routinely for all our antibodies. For Western blots, we use all 5 of them. For immunohistochemistry, it's very hard to use genetic strategy and recombinant expression because you're working with, so we use independent antibodies, orthogonal strategies. And for immunofluorescence, where we're using cell lines, we can actually use all of them, but we don't use capture MS, but you could actually. So this is the way we're using these different pillars. And the nice thing is that these are independent of each other, so you can choose the ones that are easiest for your particular application and context. So genetic strategy, it's very simple. You knock down the gene and you see that the band disappears. And obviously you can do that with siRNA and CRISPR. And it's a very easy. The problem with the genetic strategy is it is kind of hard to do it in more than one tissue or one cell line. So you're, well, one cell line. So you're kind of dependent on cell lines, and you cannot really move into tissues. And you also usually only do it in one cell line. Recombinant expression, some people would call that cheating, because you overexpress your target, and then you see that you get a nice thick band. And we still think it is a valid one, because it does show that you have a good antibody. Orthogonal strategies we like a lot. And that is that you compare the results of samples between an antibody application and an antibody independent, as in this case where we take the samples through mass spectrometry and then do Western blots, and then we just compare the staining with how many peptides we get in the mass spec. And if they correlate, we think that we have a good antibody. We really like this method. Another one that we really like, but it's a difficult one, is independent antibodies. Here you have to have antibodies which are then directed to different epitopes of the target protein. And then you just compare the results between the two antibodies or three antibodies. It's also a very nice method, but you have to have antibodies to different epitopes, and this is not always that you know that. Capture MS, the way we do it now is that we run our samples on a SDS gel. We cut out 50 then slots, and then each one of them is moved into a mass spectrometry, and we see where are the peptides for your target coming up in the mass spectrometry, and then you compare it then with the staining in the Western blot. The good thing about this is that you can use one cell line or tissue and only do the mass spec once, so you can even publish it so others can use it, and then you can use it all the time to validate your antibody. And we're quite happy with this way of doing validation. So right now, in the version that will be released in November of the Protein Atlas, we have validated actually more than 10,000, but 7,000 of the antibodies on Western blots. And they are, I'm not going to go into the details about that, but each one of them, we then actually have an antibody information page. Well, I will come to that. So it's kind of interesting then to compare these validation strategies with each other. And it's really about two different things, throughput and specificity. So when you look at specificity, we like, of course, very much the genetic strategies. The capture MS doesn't give you very good specificity. The orthogonal strategies can give you very good specificity, but not always. But the problem with genetic strategy is it's relatively hard, and it takes a lot of time, so you usually can only do it in one cell line. While others like capture MS and orthogonal, you can do a very large throughput just once and then use that for validation of many antibodies. And this will be published in Nature Communications. I think it's going to be published actually next week. So if you're more interested in this, there will be a paper coming out by Edfors et al. So right now at the Protein Atlas, we have an antibody information page. It's called Antibody Info. So what we try to do then is to have the antibodies side by side. In this case, we have 4 antibodies to this protein here. we can then compare them, and if you get the full circle, it means that it has been enhanced validated. So if you then go into more details here, you can see that it has been enhanced validated for ISC, ICC, and Western blot for 3 antibodies, and 1 antibody is only enhanced for ISC. So, and then you can sort of compare them. So what we have done then is that we have now, this is actually the progression of the Protein Atlas since the first version in 2005, and then we kind of released the number of genes and the number of antibodies, and you can actually see now that we kind of reached a plateau here, where, because basically we now have antibodies and information about all human genes and protein-coded genes. You can also see that it has kind of plateaued when it comes to antibodies, but this doesn't mean that we are sort of stopping. We're constantly replacing what we think is not so good quality antibodies with better ones. And also you can see here in green that in the version 18 we have now almost 12,000 proteins which are antibodies that are validated with this enhanced validation, and there will be even more now in the version 19. So in summary, the Protein Atlas, we are adhering to the antibody validation scheme outlined by this international working group. The long-term goal for us is to provide spatial protein profiles for all human proteins and only using antibodies that has been validated by at least by one of the IVGAV pillars, the 5 ones. And then just to summarize, this is very exciting for us. We're now getting antibodies also from other providers, and we're very interested in getting good antibodies that we can put into the Protein Atlas for the ones that we don't have so good quality. And then just remind you that we and the funders have decided that all of this data is open access, also including all the validation and the data. So you can go in and judge for yourself if we have done a valid validation. We're very happy. We don't have a single crown from the government. It's all funded, the whole project, by philanthropy, and we're extremely grateful for all the philanthropes that are giving us funding, not only for the Protein Atlas, but also some other efforts. The Wallenberg especially Foundation, the Novo Nordisk Foundation in Denmark, the Erling Persson Foundation, the HM owner. So when you buy clothes to HM, you're kind of supporting the Protein Atlas, which is nice. The Heart and Lung Foundation. And we also have funding from Elixir, which is this organization to share data in Europe. And with that, thanks to a lot of people and a fantastic team, and then just show you where we are in Stockholm. This is the Science for Life lab, where I live. This is the Karolinska Institute, and this is the most expensive hospital ever built. And it's not working. Okay. Thank you very much.
Well, thank you, Mathias, for a very beautiful and very informative talk. I'm very happy to open up for questions from the audience.
So I have a couple of questions concerning genetic validation. So I mean, the knockouts are kind of considered the gold standard, I think, at the moment, especially since they have become rather easy to do now with CRISPR. What do you do about housekeeping genes that cannot be knocked out? What's the percentage of those?
Yeah. So first of all, it's not gold standard for us. It's one of 5 strategies. And what we actually outline in the paper is that there are some disadvantages with genetic strategies because you have housekeeping genes that cannot be knocked out. And there's actually quite a few proteins that are actually, when you knock them out, a lot of things are happening. So that's why we're actually saying that in those cases it might be better to use some of the other pillars. But I should also say that one of the problems with genetic strategies is that usually you can only knock out one cell line, usually. I mean, you can, of course, go into different cell lines, but most people that use what you call golden standard, you're only knocking out the genes in that cell line. And of course, then you see that it works in that cell line, but there could also, of course, be other cell lines where it doesn't work. So I think genetic strategy is, of course, pedagogically extremely nice. You know, you have a band and it disappears. But so is with some of the others. So I think the best if you can use 2 or 3 of these methods. And I should also say, we are working a lot with immunohistochemistry, and there you can't really use knockout. Because, well, if you work with human tissues. So there you cannot actually use what you call the gold standard. So it's very good, and we put a lot of efforts into genetic strategies, and we are planning a rather big collaboration with Horizon for this reason. But it has also limitations.
I've got time for one more quick one, if that's all right, just because we are pushed.
What do the data you have, can you make any predictions of when an antibody works in IHC? Let's say an antibody works in plasma and works in IHC. I know you showed an example, but is there any trend that you can
No.
see?
Yes, it's a very clear answer. We cannot predict anything. Well, obviously, no, I don't think that. And we are using a little bit special in our case because we're using antibodies that are fragments that are produced in E. coli. And they are probably not correctly folded and so on. We're now trying to use full-length proteins and see and so on. But I think that you really have to go through the validation and sort out what is working and not.
Very quickly, very quick.
Is your capture MS in the paper that you wrote in Nature Methods? That was immunoprecipitation.
Yes, yes.
In this, I think of your capture MS a little bit more like an orthogonal method.
Yes.
It's just a different. It's the question broke in 2 different dimensions.
Yes, exactly. No, you noticed that. In the paper, original paper, it was more immunocapture MS. But we are not using that here. Here we're instead using a little bit, you could call it orthogonal, but it's very special because you're cutting out bands from a Western blot. And so we call it still capture MS. We're still discussing how good it is to do immunocapture, immunoprecipitation, and then because as you know, when you do that, you get a lot of different peptides. If you find the target on those peptides, does that mean that your antibody is actually very good? We're still debating that.